Hi everybody, thanks for tuning in on Facebook Live. I'm Thomas Detchemendy. I'm here to talk to you guys about how we run our lab on a daily basis, how we make pots, make the plants grow from the seed all the way to a mature plant, and just one of the daily activities that we do in our lab. I want to mention that I'm part of Dr. Shahid Mukhtar's lab here at the Department of Biology at UAB, and let's get started. So right here I'm standing in our soil/prep room. We have all our materials from our flats to our inserts to pots, but I'm going to show you from step one how we get to a full mature plant to work with in the lab. So first off, I'm going to grab one of our pots here, and of course plants need soil, which have the nutrients, they need water, and they need sunlight to grow. Those are the three big things. So what we want to do is we want to, sorry about the noise from the bag, we autoclave our soil. That's why it's in an autoclavable bag here, so it's sterile, everything's controlled. And we want to break up some of the soil here with our hand, always wearing gloves of course, and just sprinkle some of that soil in there like you would making a pot for anything, and just pat it down a little gently so you have a nice solid media, if you will, for the plant to grow on. From here, it's dry soil. It needs water. Plant needs water to grow. And we have some water here. This is what we like to do in the lab so it doesn't get very messy. Just sprinkle some of the water on top. It doesn't have to be soaked, just a little bit moist for that seed to sprout. You remember back in the day, germinating a seed in a bag with a wet paper towel. This is kind of the same thing, but just on soil. So once we have our soil wet, you're ready to sow our seeds. So we have a tube of seeds over here. We work with the Arabidopsis seeds. So it's very tiny. They're almost like little shards of pepper or something. Very tiny. I don't know if you could even see those in there. But what we do to start off with sowing our seeds is we get any type of paper. I like to use whey paper, so it's nice and loose, lightweight, and just sprinkle a couple seeds onto that whey paper. That you might be able to see the seeds a little bit better on that view. So from here, once you have them sprinkled, you kind of do this little method where you tap your finger and just randomly disperse the seeds all over that surface of the soil. And so from here, you would think, okay, you just go put them in the greenhouse and the seeds are going to germinate and grow and create a plant. Well, to mimic the nature of winter to spring, we like to put our plants after they've been sowed, or our seeds after they've been sowed, into a cold room. Now the cold room's way down the hallway, so we're not going to do that, so we're just going to say the cold room's right down here. So put that in the cold room. It mimics winter, so we leave it in there for about a week, maybe 3 to 5 days, maybe a little half of a week. And about 5 days later, we pull it out of the cold room. We put it in sunlight in our growth room, or our greenhouse behind me here, and we have germinated plants. So I'm sure most of you know how that works, and there we are. We have our Arabidopsis plants germinated, ready to go. So from here, we want to work with an adult plant. So these are little seedlings, these are little baby plants. So we want to transplant these into a flat. And this is just a section of a flat that I'm going to show you. Over here, when I showed the beginning, usually we work with a full flat of plants. So several inserts, so we have a full thing. But for the video purpose, I'm just going to show you one little quadrant. So from here, the same thing. We need to fill it with soil, we need to get the soil wet, and then we need to provide the plant with sunlight in order to continue to grow, because they've already germinated. We have our seedlings ready, and we just need to transplant them so they are not stressed out competing with all the other seedlings in the same pot. So the same exact thing. We want to pat the soil down. To make things go by a little faster, I'm going to come over here and wet the soil with our handy dandy sink. Bam. So just just as moist as the soil that we did for the pot for sowing the seeds, the same thing. And now what we want to do here, we're going to be really careful because we don't want to stress the plant out by breaking the root or anything. So I always kind of loosen the soil around the pot like this. And I always take the whole thing and just gently dump it on a paper towel like so. So all those roots are still intact in the soil and we don't want to hurt them, so gently break up some of that soil. And I like to use little forceps or tweezers like this. I like the curved end for some reason. Some people don't. But what I'll do is I'll just gently grab a plant like so. The root's still intact. Didn't hurt it. It's good. It's happy. And now we want to bring it into a new home. So we're going to make a little incision in the soil, if you will, with your finger. I like to use a Sharpie and go all the way down and just make little holes for the plants. But for this purpose of the video, I'm just going to do one and just use my finger. So make sure your plant's nice and happy. The soil's packed down around it. And you'll do that for the whole thing, and usually we use a full flat like I showed you earlier. So we'd have, 10 of these things to fill up with plants. So from here, this takes about maybe 2 weeks to get to this stage, the seedling stage, from the seed to the seedling, after we do the cold room and all that. But from here, we want to let the plant grow a little bit longer so it gets big so we can work with the leaves. So I'll bring you into our greenhouse where our plants, we've put them in there to grow bigger. We use it to collect seeds and just make our plants more mature until they get to that point where we can use them. So here's the greenhouse. As you can see, we've got multiple plants growing in here, all different stages. But for the example, for the video purpose today, I'm going to be talking to you all about what we do when the plants get to this stage. So you just saw them as a seedling stage in the pot. We transplanted them into one of these, into a quadrant, and now they're about a month old, 3 to 4 weeks old at this stage. And so we use these plants for a number of things, but it usually starts out with injecting bacteria into the leaf. And so our lab as a whole looks at the immunity of a plant and how it responds to a bacteria infection in nature and agriculture. So farms out there, they're always combating pests and bacteria and all sorts of infections that can take down crops and really eliminate, economically important crop production. So that's what our research is really based on, and we want to find out how can we help the plants fight these bacteria. And so to find that out, we have to inject them with the bacteria to get data to determine how are we going to fight this problem. So I'm going to show you all how we inject our bacteria. Now this isn't bacteria, this is just our buffer that we would usually dilute our bacteria in, and I'm just going to show you how we do it. So right here I have, this is a 1 ml syringe. It's needleless. I'm not actually puncturing the plant with a needle here. It's not like we're at the doctor's office. Just a needleless syringe, like a flat end. And we're just going to take some of our buffer, which happens to be magnesium chloride. It's just a buffer that we use to inject all of our bacteria in plants. So I get a little bit in the syringe here. And so what we want to do is we want to be very careful about this. We don't want to damage the leaf. If you push too hard, you might damage the epidermal cells. So I'm going to take the leaf like this. Sorry if you can't see very well. And inject the bottom of the leaf. I only did about half of the leaf there, but if you can see the difference in color from this side of the leaf to that side of the leaf, that's where I've injected the bacteria, quote unquote bacteria, and the other side of the leaf isn't injected fully. So I'm also going to inject that side. And now it should look like a translucent leaf because inside of that leaf we have injected our bacteria. And so we'll leave those plants We usually do 4 leaves per plant, so I'm just going to show you one for the video. But we'll do that, and we'll let the plants try to combat that infection, that bacteria that we just put inside of their cells. So we wait about 3 days, 2 to 3 days, and we actually use a hole puncher like you would find at your house for the next step. And so what we do is we'll take a leaf off. I'm not going to use the one I just injected because it's a little wet. Usually we'd use tweezers for this, but I don't have them they're over there. And we'll take the leaf that we injected with the bacteria 3 days later, and we'll hole punch a little leaf disc out. Now, like I said, we inject 4 leaves per plant, so we'll mix up the plants so we're not getting the same 4 leaves in one tube. And that's just for randomization, so we get good data collection. And from here, we'll grind them up in the tube. I don't have the tubes to show you because they're kind of expensive. I don't want to waste any material. But I actually did this earlier this week. And what we'll do is from each plant, so if you do 6 plants, you'll have like 1, 2, 3, 4, 5, 6. And we did 2 different bacteria, so that's why we used the whole row here. But we want to use all of the bacteria, the leaf discs that we take, we crush them up, and we dilute them down 5 times here on this 96 well plate. And we do this dilution method to help us quantify the bacteria that have grown in the leaf since we injected it. So from here, we ground up our leaf tissue that we injected the bacteria in, we diluted it, and then we take a multi-channel pipette, which I don't have to show you either, but then we spot it on an agar plate like so. As you can see, it's a little overgrown, but some of these down here you might be able to count some colonies on. But really, maybe better for the camera, I can flip it over like this and you can see the actual bacteria growth. There's my hand behind it. And what we'll do from this stage is we'll count with a little Sharpie, and we will count each individual little colony on that plate. And based on that number, if we had this dilution, that dilution, and so on and so forth, we can quantify how many cells of that bacteria have grown inside the plant after we injected it. So if the plant can combat that bacteria, we would want to see less growth on this plate. So you would have growth higher up in the dilutions. But if it grew a lot, you would have all 6 rows or all 5 rows from your plate grow. So you can tell by this bacteria, only the first 3 rows had bacteria in it. So it seems like the plant, combated the infection quite well. And now we do this for a number of different genotypes. And when I say genotype, I mean it may have a mutated gene that we can order online. If we want to focus on a specific gene in the plant, we can order that seed and use that for future research purposes. And so right here I have our regular plant that we use as a control, our wild type plant, and then a random mutant that we work with in our lab. And so based on this result, they're actually quite similar together. So we wouldn't be able to infer very much just from looking at these plates. But when you quantify everything, you count it up, and you make your graph on Excel, then you can tell differences. And so we do this method for a number of different types of plants in our lab. They're all the same species, they're all Arabidopsis, but we can order different types of that species to look and see which gene might have an effect on the bacterial growth. So we can order different mutants, try out different types of expressed genes, and I don't want to get too much into the molecular biology behind it, but that's just a brief overview of how we use the plants injected with bacteria, and we want to see how well that plant can combat the pathogen. And now, are there any questions for me anybody has?
How did you get into this particular area?
So I started off actually my undergrad in a genetics course, and I was really interested in just genetics as a whole. And I talked to my professor, and he asked me if I liked plants. I mean, sure, I like plants, and so he started me working with Arabidopsis. So I've been working with Arabidopsis for the past, about 5 years now, and I was kind of just introduced to it one day as an undergrad, and I just never got out of it. I just stuck with it, and I seemed to enjoy it. So now I'm expanding all my knowledge that I had from undergrad into a more genetic level here at UAB, as Dr. Shahid Mukhtar's lab, as well as Dr. Carolina Mukhtar. We work pretty closely together, and we study a lot of the molecular genetic mechanisms behind plant immunity. And so I kind of went from just growing plants, looking at different toxins that might hurt them in nature, to actually using bacteria, so biotic stresses that would affect our plants, and we want to find out how we can battle that bacteria-plant pathogen-like interaction in agriculture.
So yeah, so then in the grand scheme of things, what does your research actually, like, how does it, I guess, implicate in the public?
So, in a very basic way to put it, I don't want to get in too much depth about the genes and the mutants and all the mutations that we can talk of, but in the grand scheme of things, by using the different types of plants, when I mentioned that earlier, the different genotypes, we can knock out a specific gene that makes it non-functional, and we can see if that gene has an effect on the actual bacteria growth in the plant. And using a collection, a library of multiple of these gene knockouts, we can see, okay, if we knock out gene A and we compare it to a wild type plant that has that gene, which one can combat the bacteria infection better. And by doing several experiments of this over time, again and again and again, we can get more data. It's little pieces of data here, little bits of pieces here and there that can help us answer really the big question for how are we going to help agricultural farms out there combat all these bacteria infections, as well as other pathogens such as fungal infections, and Oomycete, any other plant pathogens out there. Our lab is really focused on just helping agricultural communities combat these on a genetic level. So I don't want to get in too much detail about the genetics and everything that happens behind the scenes, but for this experiment that I showed in this video, it's just based on how well the bacteria is going to infect this plant. And maybe if we have a plant with a specific change, like a gene knockout or gene overexpressed, we could infer that that gene has an impact on its immunity to that bacteria. Are there any other questions for me? Alrighty, well, thanks for tuning in, everyone. Again, I'm Thomas Detchemendy. I work in Dr. Shahid Mukhtar's lab here at UAB, and thanks for listening.